and cell-mediated. We have humoral and cell-mediated immunity. And we have seen what under humoral is. Humoral, we are looking at the production of antibodies. And these antibodies are being produced by the B lymphocytes. Whereas under cell-mediated, the key player is the T lymphocytes. And these T lymphocytes, we're going to see them being divided into two, into three, whereby we have the CD4 positive, the CD8 positive cells, and the T suppressor, isn't it? These are the ones. Whereby the CD4 we call them T helper, the CD8 we call them T cytotoxic cells. This one will do a good role in cell-mediated immunity. Then in the humoral, we are looking at the antibodies being produced by the B lymphocytes and being specific. So this one is specific in action, but this one is not so much specific, but we shall look at it. So we are going to look at the pathways in this. Of course, in this, we are going to look at the role, different pathways in acquired immunity. We are going to begin with the cytosolic pathway, isn't it? I'm saying that this acquired immunity, we can discuss it under two different pathways. It has two pathways, whereby one of the pathway in this is what we call cytosolic. Cytosolic. No, this is not permanent. This is permanent. Let's play that good. It's permanent. Let me throw it down. So we have so we have cytosolic and what? Green, though green does not look green it well. We have cytosolic pathway. Cytosolic, you can call it endogenous. Endogenous pathway. So we shall look into this. Then after cytosolic, we have also what we call it exogenous, but Rosani, can you remind me another name? Exogenous pathway. Exogenous pathway. These are the two we are going to see how they interact. All of them work hand in hand to come up with the results. So let's look at them. So I'm beginning with the cytosolic pathway. And this cytosolic pathway, the guys we need in cytosolic, it deals with endogenous antibodies. It deals with endogenous antigens. Endogenous antigens, like virus-infected cells. Virus-infected. So the antigen which is triggering this immunity is inside the cell, isn't it? That's why we are calling it endogenous. And the cells here which are involved are all nucleated cells. Any cell that is nucleated is involved in this cytosolic pathway, which we are calling This one, that's why we call it endogenous, because it deals with infected cells, tumor-infected cells, viral-infected cells, which are already inside, not in the extracellular fluid, but those infected cells already, which are already infected. And this pathway, we shall see another requirement we shall need major histocompatibility complex, and here we use type 1. Major histocompatibility complex type 1. Because this major histocompatibility type 1 is the one that is found on all. This one is found on all nucleated cells. Found on all nucleated cells. So all nucleated cells have major histocompatibility complex type 1. Sometimes some books call it HLH, human leukocyte antigen. And possibly the other man is called who? You're called stop codon. This major histocompatibility complex is different from individuals, isn't it? Each individual has a different major histocompatibility. That's why we call it major histocompatibility. Major histocompatibility. This word it has histo, which means it tissue, then compatibility means they can. So this is the one that brings the problem of transplant rejection. I think that. Even it is the one that you can bring about your blood being reacting to another. Because every different person has a different. And this major histocompatibility is manufactured on chromosome number 6. Chromosome 6. And it differs from individual to individual. It is hard to find mine resembling yours. So they differ from individual. That's why they are calling it major histocompatibility. So for you to donate the kidney to me, your tissues must be compatible with mine. Meaning the major histocompatibility should be matching, isn't it? Then lastly, we shall need this major histocompatibility complex type 1 presents the peptide to CD8. CD8 positive cells. Those are what we call cytotoxic cells. So that whenever the viral infected cell inside the cell internalizes the peptide, as we are going to see, then we present a portion on top. On major histocompatibility, then the cytotoxic are triggered to come and what? Start killing that cell. As we are going to see the pathway as it moves. So let us begin the process. That was just an introduction of it. So the process moves like this. Hope you have them in the notes. Should not give you the ones you don't have. So we are saying this process of cytosolic pathway. This cytosolic pathway, I want to draw a nucleated red cell. You know, a nucleated cell. So if I have a nucleated cell like this, it has a nucleus. This is its nucleus. Of course, this guy will have rough endoplasmic. How do we draw rough endoplasmic reticulum? I can just present this one as rough endoplasmic reticulum. And this cell is going to have our major histocompatibility complex, as we shall see. But what we are saying that this nucleated cell is infected with a virus. Maybe let me say this is a virus. This is a virus. So what happens is this virus, it undergoes, we know in the cytoplasm we have lysosomes, which are proteases, isn't it? So those proteases will come and break down. We shall need the action of proteases. If I can zoom it here, is that if I have a virus antigen, then plus a marker, we need something to come and mark it. And that guy is called ubiquitin. Have you ever heard of it? Fahim, have you heard of ubiquitin? Ubiquitin is a marker. He marks the what? The antigen, isn't it? So that the proteases can come and only attack the marked virus, not your own body cells, isn't it? So ubiquitin is labeling the virus so that when the proteases come, will only go to the affected cells. So in the presence of proteases, and sometimes we need energy in the form of ATP, we are able to break down this virus into peptides. And these peptides are always between 8 to 10. So what it does, these proteases have scissors in their hands. They come and start breaking down this virus. And this virus will form small what? Peptides. So these are peptides formed. From peptides, you can allow me the nucleus to put it up so that it doesn't disturb me. This is the nucleus. So after forming the virus, which was complete, now we have broken it down into smaller portions. So we are going to pick only one portion. We present it to the what? So as we are here that within rough endoplasmic reticulum, we are going to form our major histocompatibility complex type 1. Is in the rough endoplasmic reticulum. This, we are going to transport, actually the major histocompatibility complex is here. So what happens is there is a transporter, stop codon, there is a transporter that we shall come and we get one peptide, we put it on the major histocompatibility class 1. So we get one peptide and we put it there. We form what we call histocompatibility type class 1 peptide complex. We can call it peptide major histocompatibility type 1 complex. Then it is this complex. These things are in your notes, isn't it? They're in your notes. So we are going to transport this into rough endoplasmic reticulum. If I can put rough endoplasmic reticulum this side, this is my rough endoplasmic reticulum. I'm going to transport this complex inside, isn't it? So we shall have our major histocompatibility here with its peptide viral portion. Why are we, and this one is transported by a guy called the TAP. Transporter antigen protein is the one that transports it inside there. Say this. Transports it inside the endoplasmic reticulum. Then later, this guy, so long as the immune system is triggered, it comes and presents it on the surface. So we shall get our major histocompatibility complex type 1 on the surface of the cell. This is our major. It is presented on the surface with our peptide. Is presented. And whom do they present it to? Is the question. Target, whom do they present it to? They are presenting it to CD8, isn't it? Which are T cytotoxic cells. So you'll find here on the top I have the CD8. Let me draw it here. So this CD8 has also its receptor. And this receptor, we call it TCR, T cell receptor. And it has a hand which strengthens this, and this hand is the one that is called the CD8 portion. So it uses the CD8 portion to come and interact with the virus, which is presented by major histocompatibility complex type 1. Then after coming into contact, that's when it will trigger the cytotoxic T cells to become activated. Now actually this is the CD, the whole of it, we can call it the CD8. And this CD8, once it interacts with the peptide, it starts producing what we call cytokines. And these cytokines are the one that will activate. It will activate this T cytotoxic cell, and this T cytotoxic cell will come and kill the infected. So this one marks the end of the pathway. That is the end of the pathway. So what are we saying? We are saying in summary, we have a virus, isn't it, which has infected our cell. We need to break into smaller portions so that we can present some. So using proteases in the cell, in the presence of ATP and ubiquitin as a marker, we've cut it into smaller portions. And these peptides are put on the major histocompatibility. Because major histocompatibility type 1 is also manufactured within the cell. So when they are coming out by exocytosis, they come together when it is already with the peptide portion, what you call peptide MCH type 1 complex, isn't it? So it presents the portion of the virus, and now what happens is that the CD8s come and starts being activated. And what activates them is that these cytokines, like interleukin, we have interleukin 1, interleukin 12, and the tumor necrotic factor gamma. Is it gamma or alpha? We have, no, it is called interferon gamma. Interferon gamma. So these interferon gammas are the ones that come and act on the same cell after producing cytokines, come and activate the same cell which produces them. You have heard of what we call those enzymes, those hormones which were acting on the same cell that are producing them. They were called what? They're called what? Those hormones. You find the cell that is producing a hormone. The hormone acts on the same cell that is producing it. No, they are not in here. Autocrine glands, isn't it? Autocrine glands. So this one is acting as like an auto. It produces these chemicals, cytokines, like interleukin 12 and interferon gamma, which come and activates on the same cell. To activate it to come, it comes and clears the whole cell. So that even the virus which is inside is also killed. You remember these viral infected cells, tumor cells, are supposed to be cleared from the saturation. So these cytotoxic come and kill the whole cell together with the virus inside. So that is the only pathway which we call cytosolic or endogenous. So the major points to get is that after knowing this process, you must know that it occurs only in antigens which are found within the cell, not extracellular. And that it occurs on all nuclei. And the key player is major histocompatibility complex type 1, which presents the peptide portion of the pathogen on the surface to the CD8 so that it can easily kill it. So that this CD8 produces cytokines which activates the macrophages. Apart from activating this cytotoxic, this interferon gamma also activates macrophages. And sometimes it can also activate the natural killer cells. So all of them, these three, the cytotoxic, the macrophages, and the natural killer cells, are the ones that come and clears the whole infected cell. But who is triggering them is the cytokines, the chemical mediators that are produced after being presented with the peptide portion. Because these guys must come when they are specific to what they want to kill. Which cell is? The macrophages in the liver are called the Kupffer cells. Yeah. That is the same. So long as it is a virus and it is producing the antigen, of course the antigen is now the virus, and it is one virus having all those different proteins which are antigens. So all of them are presented at once by the major histocompatibility complex type 1. Because it is the same component. So that is the pathway. This one, we are keeping them. You see, when they remain, if it remains here redundant in the cytosol, it might start also triggering other, it can lead to autoimmunity. So we are taking it to keep it safely. So that whenever we need it to present, it comes out and presents. Because we don't want to leave it there, it will keep activating our cells. Isn't it? And it can lead to what we call autoimmunity. So we preserve it so that when we need it directly, it is presented on the surface in the presence of the CD8. Please, do you want to ask a question? I think you are being... So in summary, there are only like three steps. Step number 1 is the formation of the peptide, isn't it? Process number 2 is this peptide being loaded on the major histocompatibility complex type 1, then transported to the rough endoplasmic by TAP, isn't it? Then the third one is presentation on the surface. I can summarize it into those three steps. Transporter, antigen, protein. Transporter, antigen, protein. The one that transports. Yes. Panji? We said the first one is the proteolysis of the antigen to form peptides. The second one was loading the peptide. I was using the word loading. Or formation of peptide major histocompatibility type 1 complex, whereby you get a peptide, it combines with the major histocompatibility to form that complex, and it is taken to the rough endoplasmic to be stored. Then when needed, it is presented on the surface. Those are the major 3 steps I want you to know. TCR is found on the cytotoxic, on the CD8. And TCR is the one that interacts with the peptide to know which cytokine to produce, isn't it? So after proteolysis, breaking down the virus into peptide, we load it on the major histocompatibility complex. Then we transport it by TAP into the rough. The third one is presentation to the CD8. You are presenting the peptide on the major to the CD8, isn't it? After presenting this, after it interacting with the peptide, now it will produce cytokines, which will come and activate the same cell. It can kill. It can even go ahead and activates the produces the interferon gamma, which activates macrophages and the natural killer cells. And the role of natural killer cells is to come and clear the cell, the infected cell. This MHC is helping us to present this capeptide to the CD8, isn't it? That is its role. Because without this peptide, you cannot activate the cytotoxic to start killing, isn't it? No, we are presenting MHC1 presents a peptide to the CD8. Are you getting that statement? But we are not presenting the MHC. The MHC is the one presenting the peptide. And apart from cytokines, before I receive a question from Peace, there are other cells which are, there is what we call apoptosis, or what we call programmed cell death. Now these guys can produce other chemical mediators, chemicals like perfluorins and granzymes. And these perfluorins and granzymes, the name you hear it, that perfluorins will go to the cell and starts creating what? It perforates, creates pores, leading to apoptosis. So these guys, for them, they trigger what you call apoptosis. And the granzymes, they are produced also by the cytotoxins. That if it is not cleared well, we can induce apoptosis. And there is also, that process is also very big, apoptosis process. But majorly is that in case we have not cleared it, we can induce apoptosis by the perfluorins and the granzymes. If you want, you will read about it more, how it comes about by the apoptosis. Cytokines. After this TCR are interacting with the peptide presented by MHC1, it produces the cytokines, it produces the perfluorins, the granzymes. Cytokines, we have example like interleukin 1 and interleukin 12 and the interferon gamma. And these ones we have said, these activate it comes and acts on the same cell that is producing it to activate it. It also activates the macrophages and the natural cells, that they come and clear wholesomely the whole infected cell. If it is a tumor, that's why we don't develop cancer when you're immunocompetent. The moment you get a cancerous cell, this process comes in, isn't it? We produce the cytokines and the interferon gamma, which activates macrophages and the natural killer cells. They come and clear the tumor-infected cells. When you are that's why you see cancer is common in immunocompromised. If you go to Cancer Institute and you make a survey, you find the common cancerous patients are HIV patients. True or false? Most of them. But I'm not saying 100%. Most. Even the normal ones are there. Keeping factors constant. Maybe you never know you have married a woman is always stressing you. Of course, stress reduces immune. So when a tumor cell comes in, it survives. Isn't it? So even stress. Stress can also do that. Even when you don't have HIV. So not HIV, it is not only HIV that lowers the immunity. Even any factor that lowers immunity. To get cancer, your immunity must be low. Otherwise, if this process is working well, that cell which becomes cancerous is cleared. It undergoes either apoptosis or the natural killer cells come and clears it from the circulation. But once your wife is stressing you, You are no longer sleeping. Trust me, cancer can come in. Yes. Mm. 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 I don't know how does all this happen. Mm. Yes, the bird can this cell. Then how does this cell I need to have a specific cell that I will not get the question. I mean, for in a mm. Against a virus, how do we cell to know that this is a virus, a specific virus? Especially the CPH, how does it feel like of course, before they are released into saturation, when they go into saturation, they are trained. But that one can be answered well by the humoral immunity. Not the T. But still, your question is that how is this CD4 knowing that this is for HIV? I need to have this kind of receptor to react on that complex. No, the receptor is TCS, T cell receptor. No, it is not specific. It is not specific. For all. So long as, even though when this is a tumor, not a virus, still the same process. Because just you are going to clear. It is like if I know that Adu is in this complex, what they do, we throw the bomb on the whole complex. Adu can even die with the other normal ones, the innocent ones. Even the ones who are innocent can die with the Adu. That's what happens. But for the body does not do that. It only goes to the infected. It selects the only infected and kills it. Whether it is a virus, whether it is a tumor, the same process. The major thing is producing cytokines which activate it. Because without any presentation, it is naive, it is not active. So that's why we need to be active, need to be first be activated by presentation of the major histocompatibility complex. So this is the end of cytosolic pathway, or what we call endogenous pathway. And up to now, even in your body, this process is happening, but you don't know. Can you sit inside your body and you see? It is happening. It is happening. By of course enhancing your immunity. Immunity. That's why we normally say when you come and we do, your neutrophils are low, your lymphocytes are coming high, we give you vitamin B complex. Those immune, in COVID we were giving vitamin C, just to activate the immunity so that it is strong. Can I rub this? So this is the side of endogenous.